All right. Can you guys hear me okay? Like Dr. Jane said, my name is Dan Ababayu. I'm a postdoc in Tom Barker's lab, and I'm really happy to be here today. 11 years ago, I was where you were sitting. It was not taught by Dr. Janes, but I can still remember the pain and frustration with the first quiz. So I challenge you guys not to focus on the things you got wrong in the last quiz, but to try and focus and get ready for the next quiz. So today we'll be talking about cell membranes, and we'll be couching that within the context of the disease lupus. So lupus is an autoimmune disease associated with antibodies against nuclear proteins. So does anyone want to take a guess at defining what an autoimmune disease is? Yeah. Yeah, so exactly right. The best way to describe it is that your immune system is recognizing self as foreign. And so that can be recognizing either proteins or cells. And so the antibodies that are present here are made by B cells, and they recognize nuclear proteins. So normally antibodies are recognizing things outside of the cell, but the nuclear proteins are becoming accessible to these antibodies. And so that's part of the explanation for this disease. Some of the symptoms are fatigue, joint pain, and photosensitivity. And in addition to that, there's a malar rash and anemia. So the malar rash is basically like a butterfly rash across your face. And another thing is, does anyone know what the word lupus means, or the root of the word lupus? Yeah, wolf, that's exactly right. So the idea is that back in the 13th century, there was a doctor that first saw a patient present with lupus, and it resembled a wolf bite. So there are a lot of things in medicine that have a weird origin like that when it's not actually having anything to do with a wolf, but it just has its origins with a story like that. So the lupus is also referred to as the great imitator, because if you look at these list of symptoms, these are largely systemic, broad symptoms that can sometimes be attributed to, that can sometimes be present in other diseases. So it's sometimes really hard to diagnose lupus because these symptoms are fairly common in other diseases. And so this disease alternates between flare-ups and periods of remission. Does anyone want to give me a definition of what remission is? Yeah. Yeah, exactly right, when the symptoms are no longer present. So just for a little context, I want you guys to feel free to ask me questions, because I can remember being in your seats and being confused and not understanding things very well, and I also will ask you guys questions as well, just to try and make this as informative or interactive as possible. So, looking at the prevalence and the incidence, the prevalence for lupus is estimated at 1.5 million people in the U.S. and 5 million people worldwide. But given that lupus can be so hard to diagnose, there might be far more people in our nation and globally that have lupus but are misdiagnosed. And so women are unfortunately 9 to 10 times more likely to develop lupus than men. That's largely pretty true across most autoimmune diseases. There seem to be some traits or some genes that are carried across women and not in men that predispose them to autoimmune diseases, and that's true here with lupus as well. And that's also particularly true with non-Caucasian women, who are 2 to 3 times more likely to develop lupus. When you look at the cost of lupus, it has $18 billion in direct expenditures. Does anyone want to define what direct expenditures are? Or posit a guess at what it is? Yeah? Yeah, so costs with treatment, with therapies, and the indirect costs are all the things that are associated with loss of income, the secondary effects. And so the direct expenditures are treatment and therapies. So the etiology of the disease. Does anyone want to help me define what etiology is? Yeah. Yes, exactly right. Cause. Another word for so the cause of lupus is unknown, or another word for saying that is idiopathic. That's a large word that's oftentimes used in medicine to say we don't know what causes this. So, for example, in Tom Barker's lab, I study idiopathic pulmonary fibrosis. Fibrosis is scarring, and so I study scarring of the lung where we don't know the cause. There are some instances where we do, but the one that we're most interested in is this one where we have no idea what the cause is, and it's incredibly deadly. So the etiology of lupus is unknown. There are genetic and environmental factors that are at play, and historically people thought that the environmental factors were what caused the disease, but that has proven to not be the case. But it does exacerbate it. It does make it worse. And so the pathogenesis of the disease, does anyone want to take a guess at what pathogenesis is? The nice thing about medical terms is that you can break them down into pieces to try and take a stab at what they mean. Yeah. That's exactly right. You took the words right out of my notes. It's the progression of the disease. So the progression of lupus is not entirely known, but we will talk about one potential mechanism that deals with the cell membrane. So the cell membrane is comprised of a lipid bilayer that has phospholipids as well as cholesterol within it. And then in addition to that, there are also proteins that play a critical role in the function of the cell membrane. So what's important, so the terminology here is really important. So a lot of times it's easy to confuse to interchange lipid bilayer and the cell membrane, but those two words mean very different things. The phospholipid bilayer is a component of the cell membrane, but many of the important functions of the cell membrane are carried out by the protein. So whenever, with respect to questions or answers you provide for quizzes or tests, it's important to distinguish which one is being asked of you or which one you're responding with. So the phospholipid bilayer is comprised of 4 critical parts. The first part is the fatty acid chain. So the fatty acid chain is comprised of largely hydrocarbons, and so the hydrocarbons have a ester bond that binds to the glycerol, and that glycerol is then bound to this phosphate group with a phosphoester bond. And so now the last part is the alcohol on the far right side. Can you see my mouse? There we go. I've not been using my mouse. So the last group of the phospholipid is the alcohol. And so so the fatty acid chain, the glycerol, and the phosphate group are referred to as phosphatidic acid. And with the naming of this phospholipid, it is oftentimes possessing the phosphatidyl for phosphatidic acid, and then the alcohol, whichever alcohol is present, determines what the last part of the name is. And so we have 4 alcohols that can make up, or 4 different kinds of alcohols that can make up the phospholipid. And so they're choline, serine, which is also one of the amino acids that you guys learned about in the first lecture, phenylamine, and inositol. And so usually what we'll see is phosphatidylcholine. And so the choline is the alcohol that's present here on the phospholipid present. And the alcohols, so this is important, the alcohols, there's only 4 of them, but they determine what the diversity is of the phospholipid present. And so when we look at the phospholipids, there's the fatty acid chain that we looked at earlier is hydrophobic. What does hydrophobic mean? Yeah, so it wants to repel away from water. And so when you have a phospholipid bilayer, the 2 fatty acid chains will try and repel away from the water and create a bilayer together with the polar heads on the outside. And so it's intended to try and reduce the surface area that's interacting with water. And so this is largely what the phospholipid bilayer is going to look like. And what's also important, lose my mouse, there we go, is this kink right here is created by a double bond. And so we're going to talk more about that later, the importance of that kink and the double bond present there. So far, are there any questions? Yes? So the different alcohols just determine what kind of phospholipid it is. So they all share a common prefix, phosphatidyl, and then the alcohol determines what it is. So phosphatidylcholine, phosphatidylserine. Does that make sense? Okay. And we'll talk, and actually later in the lecture, we'll actually talk more about, either if we can get to it, what the location of these different phospholipids within the phospholipid bilayer. Did you have a question? The proteins. So I'll get to their different roles actually in a few slides. But the two main components of the cell membrane that are critical are the phospholipid bilayer and proteins. Good question. Any other questions? All right. So as far as considering the scale of, or the size of these components that we're talking about, the cell membrane is about 4 nanometers thick. So that's 4 times 10 to the minus 9. So this is it's incredibly small, but bears an incredible importance for the cell's function. Proteins themselves can range between 1 to 100 nanometers. And so you can kind of tell right away that there are some proteins, like I was saying a moment ago, that are part of the cell membrane that can actually, if they're large enough, can span across the distance of the cell membrane. And so we'll talk more about that momentarily. But the size or scale of these proteins are largely determined by their mass and their shape. And so, as you guys, it looks like from the quiz that you guys were taking, you guys can understand that proteins undergo different conformations that can dictate their shape and thus their size. And a typical cell is on the order of 10 microns in diameter. And so that's average, because there's cells that do many different things and have really large sizes. But the average is 10 microns. And so just for scale, your hair, depending on its fineness, can be between 20 to 200 microns. On average, hair is usually about 100 microns. And so what's helpful for me is that if you look at the thickness of your hair, that's about 10 times as thick on average as the average cell in diameter. And so for scale, by raising your hand, what do you think the largest part of a cell is? So we know that a cell is 10 microns in diameter on average, but what do you think the largest component is? Yes. Yes, that's exactly right. It's nucleus. It's smack dab in the center, and it possesses the DNA as well as a lot of important nuclear proteins. And so does anyone want to posit a guess as to what the next largest component of the cell is? And you can kind of relate it to importance, if that helps give you a hint. Yeah. Yeah, that's right. So the nucleus is the largest part. The next part is the mitochondria. And then after that, what do you got if you need to guess, you can. What's the next largest part of the cell? Think of what kind of constitutes the cytosolic region that helps give the cell shape. Yeah? You got it right. Yeah. The cytoskeletal filaments. And so the nucleus is the largest part of the mitochondria, and then after that, the cytoskeletal filaments. So this is a quick snapshot of what the phospholipid bilayer looks like. And so this is the polar heads of the phospholipids, and so this is where the glycerol, the phosphate group, as well as the alcohol present on the phospholipid. And the inner region, the leaflets here are the fatty acids that you can kind of appreciate that there's some degrees of kinks within each of them. And so we're going to talk more about the importance of that kinking later on. But what's also important is that this isn't just a stationary structure. The phospholipid bilayer is always, or the cell membrane, I should say, to be careful. The cell membrane is not just a solid structure or immobile structure. It's constantly moving and wiggling. And what kind of helps to dictate that movement or that wiggling is the temperature. So it's important to know that the temperature can kind of help slow down or help speed up that wiggling. And so, for example, in a lab, when we want to look at cell signaling or cell receptor location on a cell, we put our cells on ice. We don't freeze them because that would kill them, but we put them on ice to slow down that wiggling so that these proteins or whatever else is on the surface of the cell we're interested in isn't moving around a ton, but we kind of try and help stabilize it to look at it better. Does that make sense? All right. So the fluid mosaic model is a really important and really helpful. Oh, yes?
So if you were to put the bilayer into like a completely non-polar solution, would it like invert itself so that the non-polar tails are facing outward and
No. So what it would do is it would help and I'll allow you to chime in whenever you want, Dr. James. But yeah, exactly. So my first response to that is that when it's in a nonpolar solution, that usually helps disrupt the cell membrane a little bit and help get things through the cell membrane. For example, like steroids are easier to get in, or any kind of drug is easier to enter in through the cell when it's in a nonpolar solution because it can help disrupt the cell membrane.
I think repeat your question if I'm sure that I understand it properly. You're asking if you were to take the same phospholipids and take all the water, or 99.9% of the water away, and replace it with pentane or a hydrocarbon or something, would it be backwards? So the question is, and in that setting, not at all physiological, but thermodynamically possible. The answer is yes, it would. Oh, and this is a good reminder. Josh had reminded me. What I or Dan write on the board would not go on the recording. So if you take particular attention to that, that's fine. These are the hydrophilic heads. It would form what's called an inverted micelle, so H2O in the middle. Any residual water would be on the inside heads, and then all of the tails will be out in with the hydrocarbon dissolved in them. So biophysically possible that aren't encountered in biology, but the biophysical principles of the membrane hold for carbon. They don't very hard part of the cell or not.
So to help better understand the cell membrane, we use what's called a fluid mosaic model. So to break that down from the different components of that, the fluid portion of the fluid mosaic model comes from the lipids or the phospholipids being able to move and flow in the bilayer. So like I was saying before, it's not a stationary structure. It's a structure that can move and flow. And so the fluid-like nature of it comes from the phospholipids, but particularly the fatty acid portion of the phospholipid. So the fatty acid tail, the fatty acid chain, helps facilitate that fluid-like movement. And the mosaic portion of this model are the proteins that are within the bilayer. So the proteins can sit on top, can be embedded, or go across the entire membrane. We'll talk more about that in a bit. And so the phospholipid bilayer is largely just a physical barrier, but it does help serve an important function in helping to maintain concentration gradients between the extracellular and intracellular portions. But the proteins can help serve different purposes based on where they're located. So there are peripheral membrane proteins that sit on top of the cell, or the cell membrane. And so the means by which they sit on top of the cell or that they're binding to, do you guys want to take any guess as to what kind of bonds exist there between this protein that sits on top? Yeah, exactly right. Hydrogen bonds, ionic bonds. And so whenever we want to disrupt these cell membrane proteins that are peripheral membrane proteins, we use salts to disrupt that ionic bonding to try and investigate them a little bit more. Next are lipid anchored proteins. So in the Golgi apparatus, a lot of times proteins can be cleaved and have a lipid tag to the protein. And so in our lab, we're really interested in Fa19, which is a lipid anchored protein. And so the lipid anchoring of it helps confer some function to that protein, where there's downstream signaling that's unique to these lipid anchored proteins. Does that make sense? Kind of, maybe? All right. And lastly are integral membrane proteins. So one way in which it can be integrated into the membrane is that it kind of shoves itself down in there, and it's able to exist there because that surface that is present right in the leaflet of the phospholipid bilayer, surrounded by these hydrophobic hydrocarbons, is that the surface here of this protein is hydrophobic. And so it helps it to, it helps it to exist there, whereas most other proteins wouldn't. And lastly, we have transmembrane proteins that can traverse across the entire length of a cell membrane. And so there are different kinds of transmembrane proteins that serve different functions, but the nature of their amino acid sequence allows them to exist across the entire length of the cell membrane. And so, like we said before, the fatty acid chain of the cell membrane allows there to be fluid flow and some diffusion laterally across the cell membrane. Now, if you tried to take this transmembrane protein and pull it out, that would take an incredibly high amount of energy. But because of the thermal energy we have from temperature, there is allowed to be some lateral movement in the lateral plane of the cell membrane. And so for the integral transmembrane proteins that traverse across the entire length of the cell membrane, what's important for it being allowed to do that are these hydrophobic alpha helices. And so the transmembrane helices, largely each turn is, where's my mouse? There we go. Is about half a nanometer in length, and there's about 3.5 amino acids per turn. And so what's important to know is that if the cell membrane is about 4 or 5 nanometers in 4 or 5 nanometers in length or in thickness, there's a certain required amount of amino acids necessary to go across that hydrophobic cell membrane. So, to test your knowledge, if we have a 4 nanometer thick cell membrane, and if it's 3.5 amino acids per turn, and each turn is half a nanometer, how many amino acids would we need? How many hydrophobic amino acids would we need to stretch across the cell membrane? So you guys can do a quick back of the napkin calculation, and I'll give you a few seconds to do that. But yeah, so a cell membrane is 4 nanometers in length. Each turn of the alpha helices are half a nanometer, and there's 3.5 amino acids per that half nanometer. So how many hydrophobic amino acids would you need to go across the cell membrane? All right, so is anyone ready for a guess? Yeah? Yep, 25 is right. So because the cell membrane is 4 nanometers and each turn is about half a nanometer, you'd need 8 turns to make it across, about 8 turns to make it across the cell membrane. And if you have 3.5 amino acids, it's about approximately 25. You can say 21 to 28 is probably like a more exact range, but 25 is right. So one of the questions in the field right now is how can we identify these membrane-spanning segments of a protein's amino acid sequence based purely on its primary structure, based primarily on its amino acid sequence. And so what we can do now is we can bioinformatically go in, look at the amino acid sequence, and be able to determine which proteins are likely to go across the cell membrane and what parts of it can go across the cell membrane. So this is a hydropathy or a hydrophobicity plot. And so what this does is that it goes across the entire length of a protein's amino acid sequence, and it's able to measure the degree of hydrophobicity of that protein based on its amino acid sequence. And so the red bar in this, the red box in the middle of this bar looks at each individual amino acid but then also kind of and tries to determine the surrounding amino acids around it, how hydrophobic they are, and weights that, and then creates this plot that lets you determine how hydrophobic different regions of this protein are. And so, if you were to go across and do hydropathy, does anyone want to take a guess as to how many regions of this protein are transmembrane proteins? Or sorry, how many regions of this protein span the cell membrane, go across it? Just by looking at the plot, can anyone take a guess how many segments of it go across the cell membrane? Yeah. Yep, 4 is right. So this protein in particular is tetraspanin, and so there's actual proteins that have a structure that looks strikingly similar to this plot, where if you have your cell membrane, if this is really crude, right here, you'll have proteins that actually go up and down like this throughout the course of throughout the length of the cell membrane. And so this is a really helpful tool that allows you to determine is it a transmembrane protein, and what segments of it are membrane spanning segments. Yes?
How do the transmembrane proteins actually get into the membrane?
I'll take that as my cop-out.
I have a whole lecture on that. I return.
Any other questions? Yeah. Yeah, so the more hydrophobic it is, the more likely it is to span across the cell membrane. As long as it has, like we calculated, at least 25 hydrophobic amino acids in length. Does that make sense? Are there any other questions? All right. So the membrane functions are dependent upon the proteins associated within the bilayer as much as the bilayer itself. So the lipid bilayer, like we said, is largely a barrier, but this barrier does help to separate out concentration gradients from inside the cell and outside the cell. So I suspect most of you guys are taking cell phys right now, or physiology right now, and so no. You split the core. Man, it's different than it was 10 years ago. So what I was going to say, for those of you who are taking physiology, there are certain concentrations of ions inside the cell and outside the cell, and this helps to distinguish, helps with different functions of the cell as far as action potential, or even the depolarization of neurons. But the proteins that are present, like we said, we have a fluid mosaic model. The mosaic portion are the proteins in the cell membrane. The proteins are what are responsible for most other functions of a cell, or of a membrane. Sorry. So one of the functions of the membrane are compartmentalization. So one of the aspects of compartmentalization is localization of function. So as I suspect all of you went through the pre-lecture. In the pre-lecture, there's even membrane around the nucleus, and that helps to separate out different functions like transcription and translation. And I believe in the pre-lecture, you guys went over the lysosome formation and the role that the lysosome plays in cell behavior. So the lysosome actually has a portion that's pinched off from the cell membrane that isolates it out from the rest of the cell, because what's happening inside the lysosome, if it was present throughout the cell, that would kill the cell immediately. And so it helps compartmentalize and helps localize what the lysosome is doing there and not trying to kill the rest of the cell. Next is the raising the concentration. And so the lipid bilayer, like I said, the lipid bilayer helps function with compartmentalizing different concentration of ions that are extracellular or intracellular. But even more than that, if you look at the lysosome, it's helping to separate out the raising of hydrogen ions, or the acidity within the lysosome, and making sure that acidity isn't present throughout the cytosol. Does that make sense? So it's only raising the concentration inside the lysosome, not throughout the cell. A next function is the regulation of transport, and so that's a big part of what proteins do. That's one of the big responsibilities of proteins within the cell membrane. They help shuttle ions in and out of the cell. They help shuttle in different molecules, different proteins, and so they're one of the critical things they do is regulate what comes in and out of the cell. Another big responsibility of proteins is their ability to bind, to be a receptor that binds to a ligand. And so continuing my cell membrane here, on the surface of a cell, you'll have a protein that serves as a receptor. And so, that's not great. And so you'll have more than just one kind of this receptor. You'll have thousands sometimes, in some cases, versions of this receptor. So when they bind to a ligand, that they're made to bind only to that ligand, usually, the receptors will come together, dimerize, and help facilitate transduction of signals down into the cell. So they bind to a specific ligand, and they help facilitate inside-out signaling down to the cell. Lastly, the proteins on the surface of a cell will help facilitate cell-to-cell or cell-substrate binding through different receptors. And so cell-to-cell interactions, for example, whenever we have some sort of wound or bruising, a lot of times the bruising that we get from a hit will also include a lot of swelling, and this swelling is partially characterized by immune cells going to that region, and these immune cells will stop at that site because the cells in your blood vessel will bind to the immune cells, catch them there, and help them go to wherever that wound site is. And for cell substrate binding, our cells exist within a matrix, and so our cells have these proteins on the surface called integrins that will help bind to the surrounding matrix of the cells. Does that make sense, you guys? Does anybody have any questions? All right. So the membrane fluidity. So if the membrane becomes too solid, the membrane proteins cease to function normally. So, like I said earlier, the movement of the fluid-like nature is important. The fluid-like nature comes from the fatty acid chains in the phospholipid bilayer, but they help facilitate protein function. So, for example, when I talked about receptors binding to a ligand and those two protein receptors dimerizing, their ability to move and dimerize together when they bind to a ligand is facilitated by the fluid-like nature of the membrane. And so this is partially regulated by the degree of unsaturation in the fatty acid. So does anyone want to take a guess at what saturated versus unsaturated is? Yeah.
So saturated doesn't have any molecular bonds, so it doesn't have like
Mm-hmm.
and that means it has like the maximum capacity versus unsaturated,
That's exactly right. You guys came prepared today. This is great. You're making my job easier. So yeah, the degree of unsaturation is the degree of how many free, how much hydrogen bonding there is on the fatty acid chain. And so, for example, animal fat is, would you say the animal fat listed here, is that saturated or unsaturated? Yeah, exactly right. It has all of the available hydrogen binding spots filled up with hydrogen. And so olive oil is a great example of a unsaturated fat because it has that kink, and that's part of the reason why when butter is at, so let's see, so when you have butter and olive oil side by side at room temp, butter solidifies because it has a more crystalline structure that allows those fatty acid chains to line up. Whereas olive oil, because of these kinks, is allowed to remain fluid even though it's at the same temperature of butter. Does that make sense? And so these kinks are also defined as either being cis or trans. And so with olive oil, because of, where's the mouse? Because of the directionality of the side carbons from this double bond here, because they go in the same directions, that's called a cis double bond. Whereas if you look at Crisco, where's the mouse? Whereas with Crisco, when you look at that double bond, the side carbons go in different directions, so that's considered a trans double bond. This will be important later on, but does that make sense for now? Okay. And the last thing that helps determine membrane fluidity is cholesterol. We'll talk more about that in just a moment. So, as we discussed a second ago, the double bond can help determine fatty acid chain degree of kinkness, or how kinky it is. And so, out of curiosity, what pair of lipids do you think would be most ideal to have in our cell membrane or in our phospholipid bilayer? The top two or the bottom two? By raising your hand. Yeah? So if we want to have fluidity in our cell membrane, do you think the top two would be the best to have? Yeah, so the bottom two. So the bottom two the bottom two have a mixture of cis and trans double bonds, but what's great is that it provides flexibility and freedom of movement that would facilitate that membrane fluidity that we want to see. And furthermore, I'm sure a lot of you have heard of omega fatty acids or omega-3 or omega-6, or some of you take omega-3 or omega-6 fatty acids. So that omega is, what that omega means is it refers to the nth carbon, or which carbon after which you'll find the first double bond. So this omega-3 fatty acid has a double bond on the third carbon, whereas the omega-6 has the double bond over at the sixth carbon. And so cholesterol is another critical component of the cell membrane. Before I go any further, does anyone have any questions? Yes. Mm-hmm. Yeah. So when you look at the side carbons of this double bond, they're both going the same direction. Does that make sense? Okay, so for example, imagine my head is the double bond, all right, and my arms are the bonds to the side carbons. If they're going in the same direction, that's a cis double bond. Whereas if my arm is pointed down, this is a trans double bond. Does that make sense? And so if I go forward,
Maybe just the one thing to add on this.
Yes.
These alkene bonds, carbon-carbon double bond, has no rotational flexibility. It's like the peptide bond that we learned in the first lecture. That resonance structure, which is the double bond between the amine and the carbonyl, it's the same type of rigidity that's here. If you're trans, you can't just flip your arm up like that because there's a double bond between them. It gives you no rotational flexibility. That's why the definition of cis and trans is a fixed character of these lipids. Unlike the other saturated chains, those can rotate all of them. So they can move around a little bit. They can show them as that zigzag, but the position of them relative to one another does have some rotational flexibility, even though they will have to pack with one another and form a semi-solid crystal.
yeah. And so to follow up with what I was asking about the fatty acids on the next slide, between fatty acids that have a cis double bond or trans double bond, which do you think we would want in our cell membrane? I'm hearing whispers of cis, and that would be right. And so the cis double bond helps, it provides that kink that allows for the fluid, that helps, again, facilitate that fluidity we need in the cell membrane. So cholesterol is another critical component of the cell membrane. It serves to keep membranes fluid at low temperature, but then reduces that fluidity at high temperatures. So the way that it goes about doing that is if we're looking down at the polar heads of the phospholipid bilayer, and we insert a cholesterol right into the middle of that, at low temperatures, when all of those phospholipids kind of want to coalesce together, the cholesterol helps disorient that crystallinity or that degree of organization that helps make things a little bit, that provides a little bit more kinkiness to the cell membrane and helps provide more fluid flow or more room for movement within the cell membrane. Does that part make sense? So this is at low temperature, in case I didn't make that clear. So this is what it looks like when cholesterol is in there at low temperature improving the fluidity. Now, at high temperatures, because of the hydrophobicity because of the hydrophobic associations with the fatty acids. So again, this is looking down at the polar heads. If you were to go deep into it, again, we'd run into the fatty acid chains. Those fatty acid chains, again, are hydrophobic, and so is cholesterol. And so the hydrophobic association with cholesterol would keep them together and help reduce fluidity when it's at a high temperature. Does that part make sense? I'm seeing reluctant head shakes. Does anybody have any questions that I could help clarify at all? Yes. So, in the case of this is an example that comes to my mind. In the case of a fever, you have an increased temperature. And oftentimes you have, sometimes you'll have a viral infection where the virus wants to invade into the cell. And that's where it can replicate and essentially kill a target. So decreasing fluidity would help to reduce the virus invading into its target cell. Does that make sense? Yeah.
One more lethal example would be if you took a cell and you boiled it, the membrane will have so much fluidity it won't stay as a bilayer anymore. They will just wiggle out into the space and form proper micelles, so the first version of that from the cell. So excess fluidity can be no longer stay with the bilayer anymore, go out and exit the building chromosomes, lipid assembly.
Any other questions? Yeah. Sorry, can you say that one more time? What is, at high temperatures, what is it about the hydrophobic association? That is a good question. I can only take a guess, but my guess would be the change in entropy that would facilitate those hydrophobic associations at high temperatures that wouldn't exist otherwise at low temperatures. And so the energy provided by high temperatures to facilitate the hydrophobic interactions that otherwise weren't dictating what was happening at the low temperatures.
I think the one additional thing to add, chemical structure on the top of cholesterol has a polar group, hydroxyl on the left-hand side there, as well as all the hydrophobic aromatics. Similarly, hydrocarbon hangs on the right-hand side. So there are two opportunities for the cholesterol to interact with the phospholipid through the hydrophobic interaction, as well as through the hydrophilic. They have polarity to them as well. So when they line up, there's kind of two things holding them together on the right-hand side.
Any other questions? All right. So lipid rafts are regions within the cell membrane that are rich in cholesterol. And so there are these nanodomains that have where are you? That have GPI anchored proteins, that have different proteins that are created to exist within these lipid rafts, these cholesterol-rich domains, whereas other domains of the cell membrane are created to not exist within that raft. And what's important with it for these lipid rafts is that it's also seen as a signaling cluster or a signaling center. And so a lot of times, like I said, there are proteins that can go across the entire length of the membrane. And in these lipid rafts, there are certain proteins that are predisposed to come over to these lipid rafts for signaling. And so this is a kind of a signaling hub sometimes within the cell that facilitates signal transduction and downstream transcription. So, to take it all back to lupus, like we said before, for these phospholipids, there's an alcohol on the polar head, and we have only 4 different kinds of alcohols. Oh, sorry. And so in lupus, the alcohol that we want to focus on is serine. So we want to try and focus for a moment on phosphatidylserine. Now, the plasma membrane is asymmetric. What I mean by that in this context is that while the outer leaflet has certain groups of phospholipids, the inner leaflet is primarily where phosphatidylserine will be present. And so what ends up happening in the context of lupus is that during apoptosis, which is programmed cell death, so a lot of times sometimes when cells die, if you take a cell and scrape it up with a cell scraper, that's not programmed cell death. That's just mass cell death by necrosis. But programmed cell death is kind of an elegant way that cells will have these receptors and these ligands that will interact with each other, that cells will tell one another sometimes it's time for you to die. But when that doesn't happen well, it's like a game of assassins. You guys did that your first year, right? No. I said in your first year, not now. Okay, never mind. During apoptosis, phosphatidylserine will become present on the outer surface of the cell, the outer leaflet of the cell membrane. And when one of the cells that, among the cells that are phagocytes, phagocyte is eating a cell, phago eating site cell, so it's meant to eat cells. And one of the markers that helps induce phagocytosis is seeing phosphatidylserine on the outer membrane. Again, it's only usually supposed to be an inner leaflet phospholipid, but when it's present on the outer surface, that is a signal for it to eat that cell for phagocytosis. And so usually when the cells that are meant to eat the cells that when cells that have phosphatidylserine out on the surface, when they're not being eaten, when there is some sort of malfunction in apoptosis and the clearing of cells undergoing apoptosis, or as Dr. James and Dr. Barker want to put it, when the garbage men go on strike, when the cells are meant to eat them, don't eat them, what you start having is these small apoptotic blebs coming off of these cells. And so if you actually look at them under a microscope, you can actually see with certain markers, you can see that there's these like little circles popping up on the surface of these cells. And if they're not eaten by a phagocyte, what ends up happening is that they start shedding everything inside. And so we talked earlier about how in lupus, one of the potential mechanisms is that these autoantibodies, the antibodies against the self, are labeling nuclear proteins. So nuclear proteins you shouldn't be expressing antibodies against a nuclear protein, but in this case you do, and you're having an instance where they're becoming present when they shouldn't be. I think this is supposed to be a video of showing the blebbing in apoptosis, and I cannot play the media. So regardless, that was meant to be a video that helps illustrate what that blebbing looks like and what it looks like when the cells are actually shedding off their inner compartments during apoptosis. So to wrap that up, for anyone interested in trying to dive a little deeper into autoimmunity and lupus, as well as the engulfment or phagocytosis that I was referring to, we have a couple of great investigators that look into that, Dr. Kenneth Tung and Dr. Ravi Ravichandran. If you have any other questions, feel free to come on down. I'll do my best to field them. Thanks again.